we go. Oops. There it is. So we had left off talking about, like I said, the gram-positive bacilli that form endospores. Oh, no, I think I meant to pin myself here. Let's go. Let's do that. All right. And that was looking at listeria. That was, no, there we go. Clostridium. So now the non-spore forming variety are going to be generally a regular morphology, so not going to be pleomorphic. And the medically relevant ones that we see particularly in the clinic are Listeria and Erysipelathrix. Erysipelathrix. That's hard to say. Listeria is often found in dry, deli meats. And there's also a concern that it might be in things like kombucha. And we'll go into why that's the case. It's tolerant to conditions that are too harsh for other bacteria. And so pregnant women in particular are advised to stay away from deli meat, not drink kombucha, not because of the alcohol content, because it's very minimal. It's more because of this potential listeria that's lurking in there. The listeria can be anywhere from a coccobacilli, which is that kind of in between cell morphology. It's not a complete circle, it's not a complete rod, to actually elongated filaments. And while it doesn't have a capsule, it does have anywhere from 1 to 4 flagella. So it is a motile bacteria, and it's resistant to cold, heat, salt, pH extremes. And so that's why sometimes it's on deli meat, right? Because deli meat's cured with salts and it can tolerate that salt. It's kept cold, so it can still, it's not a microbiostatic control for listeria. And the pH extremes, while kombucha does, one of the byproducts is acetic acid, and that lowers the pH some, listeria can still survive in a lower pH or a higher pH. And what happens is once it enters into the host, it can enter the cell and replicate inside the cell, and then it induces phagocytosis. So it actually gets phagocytosed in order to evade the immune system. So it's like a Trojan horse, and it has virulence factors that can allow it not to be lysed by the phagosome, the phagosome in the phagocyte. So a highly adaptive bacteria. Corneobacterium diphtheriae. This is the causative agent of diphtheria. And unlike listeria, it can be pleomorphic in its shape, meaning that it has a diversity of shapes. And when you put these bacterial cells on a slide, it has this very distinctive palisade arrangement where, let me get to my little drawing tool here, right here, where it looks like, the pillars, right, of the palisades, where they kind of stack side by side. And that is a distinctive morphology of diphtheria, not exclusive to diphtheria, but one of the hallmarks. And oftentimes the reservoir that's harboring this is healthy carriers. Okay, so asymptomatic carriers. And then this will form outbreaks in children that haven't been immunized to diphtheria, live in crowded, unsanitary conditions where hygiene isn't as good as it could be. And it can be acquired via COVID style respiratory droplets, okay, from the passive or asymptomatic carriers or infected individuals. It can come from both. To treat and prevent diphtheria, there are antitoxins to counteract the exotoxins from the bacteria that it secretes, or you can treat it with antibiotics like penicillin, erythromycin. But of course, if you want to prevent it from happening altogether, there is a vaccine available that then comes with a several follow-up boosters over time. Propionibacterium. This is like Corneobacterium in that it has these pleomorphic cell shapes and can be in palisade arrangements, but it's an aerotolerant or anaerobic bacterium, and it doesn't produce toxins. What's aerotolerant? If you guys remember, aerotolerant is a bacterium that doesn't particularly utilize oxygen, but is able to exist in an environment that is oxygenated. And the most common variety is Propionibacterium acnes, which is what causes one of the major causes of acne. Okay, it resides in the sebaceous glands and it leads to the pores getting clogged with not only the bacterial cells, but the sebum that's produced and then is produced. Um, it gets more production as a result of hormones. So just going into things like puberty or pregnancy or stress that would lead to a different hormonal regulation. And then it also produces it in kind of response to the bacteria being there. So it's a double whammy. And occasionally, this bacteria can also go into the eye and interestingly is found in artificial joints. So when a knee replacement or a hip replacement is done and it's not completely sterile, sometimes this bacteria can seed into that replaced joint and cause an internal infection at that site. And I used to work in an orthopedic research lab, and I had the opportunity to go see some orthopedic surgeons do their work because I would get um, the the condyles, the femoral condyles that they would remove from hip replacements, and then the knee joints, and I would watch them, and it's a lot like carpentry, man. They got these hammers and they're banging down those artificial plates onto the bone. So you can imagine that would be more difficult to keep sterile than if you're doing a more delicate incision with laparoscopic techniques, right? Okay, moving on. Mycobacteria. This is an acid fast bacteria. And we haven't really talked much about acid fast. It's a stain. Um, and, because we haven't been able to do any face-to-face labs, you haven't done any acid fast staining. And so it's probably unfamiliar to you, but acid fast is um, used typically when bacteria don't stain gram positive or negative. In this case, um, the mycobacteria is a gram positive, but then it also stains acid fast. It's strictly aerobic, so it has to have oxygen in part of its metabolic pathway, and it produces catalase. So it's a catalase positive bacteria. And remember, catalase is one of those tests that is used to identify unknown bacteria. You run it through the gamut of gram stain, of acid fast stain, of catalase stain, of coagulase stain. And so this is a catalase positive bacteria. And of course, it doesn't form capsules or spores. It doesn't have flagella, so it's non-modal, and it's a slow grower. Okay. So these are all these general characteristics of mycobacteria. The one that you've all heard about that is a mycobacterium is leprosy, caused by Mycobacterium leprae. And so another name for this is Hansen's disease or Hansen's bacillus, but typically it's colloquially known as leprosy. It's a strict parasite, so it has to have a host to survive and perpetuate. There's no known artificial media or tissue culture that has been derived to grow this stuff yet. So that's a challenge, right? It's hard to study something when you can't grow it in the lab. Of all the mycobacterium species, this is the slowest grower, which is possibly also some of the challenges in finding a media that will harbor this bacteria. And once it does infect, it multiplies inside the cell in these large coagulated, dense aggregations called globi. And again, leprosy, as we've all heard of it, it targets the skin and the mucous membrane. So your nose is one of the first areas to go as a result of this, to necrose and to fall apart. And eventually it will progress into the nerves and attack the nerves, and then that leads to all kinds of physiological damage and malfunction, ultimately leading to death. There are endemic regions globally, so that means it's popping out. It's not isolated to just one area. Endemic means it's naturally occurring there, and we don't really know how it spreads. It's not particularly bad unless it does progress to the nervous system. And oftentimes it's associated with socioeconomic status, the living conditions, the health of the individual, whether that individual is malnourished or not, if they have a robust immune system to fight this. Because it is a slow grower, it tends to piggyback with other co-infections or an immunocompromised people. How you treat it? Well, it's a long-term treatment that really requires consistent observation by physicians and epidemiologists in these endemic areas that are at high risk, just to monitor its circulating. There's no vaccine, but there have been some trials going out there that may also align similarly with the tuberculosis vaccine, because tuberculosis is also a mycobacterium. Actinomycetes, that is a filamentous bacilli. So that means it has these long filaments that extend from the bacterial rod. And They're non-modal. These are going to be more for like attachment to the host. And so chronic infection is going to be in the skin primarily. And as you can see, these are images of the head and the upper torso having these kind of lesions form or blisters. And the two major types are actinomyces, which infects the mouth, um, the thoracic cavity, the intestines, and nocardia, which is going to infect the respiratory system and the cardiovascular system. And that's it. Oh, wow. We got through that quicker than I thought. Any questions on that, on those guys? These last couple that I talked about are the kind of the weird ones, the filamentous structures.
I have a quick question.
Mm-hmm.
Um, you said the mycobacteria are gram positive, but you use the acid fast staining to identify it.
Mm-hmm.
So it doesn't show up easily or at all with the gram stain.
Um, it so it can stain with the gram stain, but it's not as distinctive. It doesn't. So your question, the latter part, it doesn't show up as profoundly or as brilliantly as other gram positive or gram negative stains or bacteria. So it stains gram positive faintly. It's not a dark purple. It's a very faint purple. Does that answer your question?
Yes, it does. Thank
Okay.
you.
Yeah, you're welcome. And so because of that, they use the acid fast stain as another differential stain. And so that's primarily used with things like mycobacteria. Okay. And the mycobacteria, oh, what happened? That's weird. Something else popped up and kicked me out. The mycobacterium have this like waxy cell membrane or cell wall. And so it's hard to get that crystal violet through. And so it does, but it's very weak. And so that's why they do the acid fast to then further differentiate it. Okay. Let's go now. Oops, to the next one. Gram negative bacilli of medical importance. And Ryan, here's another diagram for you. So this breaks it down into the um, the fermenting aerobic bacteria. And so that's going to be this category to the left. You've probably heard of Pseudomonas. You've probably heard of Bordetella because there's a pretty common vaccine. And then we have to the far right, we have obligate anaerobes, Bacteroides. And then in the middle, we have the majority of these guys, which are the facultative anaerobes, which mean they can be anaerobic, but they don't have to be in anaerobic conditions. And so these are going to be further broken down into whether or not they ferment lactose. So E. coli, Klebsiella, Enterobacter, those are lactose fermenting. And then non-lactose, like Salmonella, Shigella, um, Serratia, Yersinia, so like the plague, Yersinia pestis, and then oxidase positive, oxidase negative. These are going to be, again, just tests, differential tests that are used to characterize and identify bacteria. So that last So when we're talking about the 6 eyes, and we're honing it down to identification, we do these different tests to then derive the bacteria that is particularly present in the sample. And had this been a face-to-face lab, that would have been your very last lab, or several labs, is to be given an unknown tube of bacteria, of culture media, and then you have to bring it through these different series of tests to figure out what bacteria it is. Does it have a capsule? So you do a capsule stain. Is it gram positive or negative? Is it streptococcus or staphylococcus? Is it bacillus? Is it acid fast? Right? Oxidase negative? Is it coagulase negative? All those things are, systematically used to get to the answer. All right. Pseudomonas. So that's a fairly common bacteria, and it's going to be small rods with just one flagella or flagellum. And so what would that be? If it's just got one little flagella, what is that called?
Monotrichus.
Monotrichus. It's free living, so it's not a strict parasite. It's found in soil, in seawater, saltwater, freshwater. Can be found on plants and animals. So it's got a very wide threshold of ecological tolerance. It's in this genera, there are very important Pseudomonas that aren't necessarily pathogenic to us, but have roles in decomposing organic matter to break it down. Some of these strains are used in bioremediation to break down, um, you know, um, uh, uh, oils and products of fossil fuels. And also happen to be frequently found in homes and clinical settings because they survive in this wide range of environments. They also have no problem coming into environments that we don't want them in. They're aerobic. They do not ferment, but they produce oxidase and catalase. Okay. So typically, you can also find a lot of these that produce water-soluble pigments, interestingly. And if you've been to Yellowstone and you look at some of the hydrothermal vents, sometimes there will be all these different colors and interesting pigments. That comes from those extremophiles that live there. And so some bacteria can produce these really vibrant colors. Pseudomonas aeruginosa, that is the bad one, particularly. Okay, that's in soil and water. It can be in the intestines of 10%, not a small amount, but residing there, not really causing any problems. But because it's resistant to some of our germicides like soaps and dyes, as well as disinfectants and antibiotics and desiccation, it's highly resistant to killing. And so therefore it thrives on things, again, that we don't want it to. And so this is the bacteria that leads to nosocomial infections acquired in hospitals, right? It can be contaminating in the ventilators that are used. It can be contaminating in the IV solution that is used, in the anesthesia equipment, in the surgical equipment. And because it can be in our intestine and be okay, that makes it an opportunistic pathogen. So in the right setting, it can be bad. And in the gut, it's not particularly great because it's taking up, space that a more commensal bacterial species could be acquiring. And oftentimes you'll see these lesions on the outside of a hospital patient that's acquired this Pseudomonas infection. Anytime you have a patient that has come in with burns, again, their epithelium, their skin, their first line of defense has been compromised all over. It's not just a little cut; it's a wide expanse of their body. And so, because the Pseudomonas is there in the hospital just waiting, it's often something that will infect burn patients. This can lead to things like pneumonia, so it can target the lungs, UTIs, so it can target the urinary tract, it can lead to abscesses on the skin, otitis, so the eyes, corneal disease, eyes. So it has a wide range of tissues that it can damage. Endocarditis can infect the heart, meningitis, brain. And so it's a pretty nasty disease. It's a pretty nasty bacteria that leads to pretty nasty diseases. It's resistant to a lot of different antibiotics, and it has this characteristic greenish blue pigment. As I mentioned, it has this pigment, right? And so these are different drugs. And so this is similar to the disinfectant lab that you guys did. Instead of having little discs of disinfectant, they have discs of antibiotics. And then this right here is the zone of inhibition. And you can see that certain drugs have different degrees of efficacy in treating and killing this bacteria. So the one I just circled, that one and this one, that has a large zone of inhibition. And then it varies. So here that has just a very narrow zone of inhibition. This one too, that is a very small zone of inhibition. And then this one is entirely resistant to this antibiotic that has no zone of inhibition. And so all the green there, that's the bacteria. And then the yellow is where that's the auger. That's where the bacteria is being repelled. Okay. Now the aerobic gram-negative rods. This is going to be Burkholderia, Acinetobacter, and Stenotrophomonas. Another mouthful. Similar to Pseudomonas, in that it has a wide variety of different environments that it can thrive in. It's an obligate aerobe, so it has to have oxygen. It doesn't ferment sugars. It's motile. It's oxidase positive, and like Pseudomonas, it's opportunistic. Okay. So these gram-negative bacteria, gram-negative Bacillus bacteria, have these similarities among themselves of being widely dispersed and opportunistic. Also, helpful in certain contexts, right? When you have an opportunistic bacteria, chances are it's doing something that is beneficial. Same with opportunistic fungi, which we'll get into, after the break. It's breaking stuff down. It's contributing to nutrient cycling in the environment. However, when that jumps ship to you, then that becomes the problem, right? Burkholderia is going to be found in the respiratory tract, the urinary tract, skin, also drug resistant. So very similar to Pseudomonas. And then these are two different species. Pseudomallii, that's going to be acquired via some kind of injury to the skin or inhaling it from an environmental reservoir like the dirt, or swimming and getting some of that water into your lungs. leading to wound infections and respiratory infections. And if it goes too far, it becomes septicemia. The patient goes septic, widespread systemic infection. Okay. Stenotrophomonas, again, water, soil, can sometimes be found in the proximity or on the roots of plants. And so because of that, because we eat plants, and with that we eat what's on the plants, and if there's a little dirt on the plant, oftentimes it's found in fecal samples from humans. So I'm not quite sure how prevalent it is in the gut microbiome per se, but it is found in fecal sampling to get an idea of the gut microbiome. And remember, when you're sampling feces, you can't just say whatever you find in that fecal sample is going to be a resident in the gut, right? There could be just things passing through. Interestingly, this is found in hospital and medical settings, not because it can't be decontaminated, but because it could be present in the agents that are used to decontaminate. Okay. So this stuff is tolerant to the disinfectants that are used to clean certain medical equipment. And it forms biofilms. And remember, the biofilms are these networks of bacterial cells that secrete this thick film and use quorum sensing to communicate with each other. And it's very hard to eradicate biofilms. You got to break through that film to kill the bacteria. And if you start, perturbing it at one spot, it can send messages to the other bacteria before that disinfectant can get there, and they can start laying out more of the biofilm protection. And so, particularly bad when the thing you're using to disinfect something is actually what is causing subsequent infection. Okay. So that's the little trivia fact to keep in mind in talking about Stenotrophomonas. All right. Cancer patients are immunocompromised and are being treated with chemotherapies, and they can be subject to these types of infections. This is one of the most drug resistant bacteria also. Okay. That's why it can hang out in our disinfectants, because it's just very tolerant. And so it has to use a special drug combo of trimethorphan and sulfamethoxazole to kind of to knock it down. So this is something that isn't really on the radar of, we think of MRSA a lot as being the bad one, but this is one to keep in mind, right, and not forget about, because luckily it's not as pervasive as things like MRSA, so Staph aureus, but who knows in time what could happen. Brucella, this is going to be a coccobacilli. So if you look here at the image, it's kind of hard. Like, that's not really a circle, and it's not really a nice circle. My circle is about as good as that. So don't look at my circle. It's not a circle. It's not a rod. It's kind of like in between, right? Kind of weird. Strepto in some cases, right? Or diplo in some cases. And these are found in livestock. Brucella abortus can lead to instantaneous fetal abortion in cattle. If the mothers are infected, it can lead to just like a spontaneous abortion. So it has an impact on the economy of farming, and Brucella suis, so pigs. You can get that from the name there, the species name, and brucellosis or Malta fever or bang disease. It's got all these different names, right? But that is the name of a disease that comes from an animal to us. Okay. So again, a zoonotic transmission. Okay. It's associated with fever that can come and go for a prolonged period of time. And luckily is treated with a combo of tetracycline and rifampin and streptomycin. There is an animal vaccine to treat the animals to prevent them from getting it, which then prevents them from losing their young, also prevents them from transmitting it to us. But in some circles, this is being considered as a weapon for bioterrorism, because you can easily take out the animals that feed the population, and then you can take out the people that feed on the animals. So when considering which microbes to use as a bioweapon, oftentimes it really comes down to dispersal. Like, how can you get something to aerosolize that will impact a large population? And luckily, that's really what kind of keeps bioterrorism at bay, is that it's hard to get these agents to disperse widely enough to have a significant impact. You can mail anthrax in envelopes to people, but that's taking them out one at a time, right? So considerations, not consideration. I mean, I'm not saying that you guys are trying to come up with bioweapons. I'm saying, be thankful that aerosolization and distribution is an issue. Not that we're completely protected by any means, though. Tularemia. Yeah, that's a good one. That comes also from zoonotic sources, particularly rabbits. Okay. It can be through contact, whether we ingest water that they have come in contact with, or even dust, right? Dust contains all kinds of things, right? I've heard, I don't know if this is true, but I've heard that most of the dust in our house is dead human skin cells, which seems kind of gross. But again, these things are inhaled. So it doesn't have to be, it doesn't have to come from a bite from an arthropod vector. It can come just from being in contact with infected animals, whether it's eating them, being bitten by them, or just being in proximity to their feces, and dander. Okay. Symptoms: headache, backache, fever, chills, just overall feeling like dog do, and just low energy, weakness. It can lead to ulcers. So the headache, the backache, those things are symptoms, right? Ulcers, swollen lymph glands, sore throat, a range of different general malaise. Again, 10% death rate. So not something to mess with, right? 1 in 10 people are going to die if they get this. Luckily, it's treated with gentamicin, streptomycin. But because it's an intracellular parasite, it can evade these antibiotics because it's protected inside the host cell, and then lead to a relapse later on. Okay, kind of goes under the radar, hangs out for a little bit. Once the antibiotics kind of work their way through and the immune response has calmed down, they can reemerge. No vaccine, and considered one of the most infectious bacterial species that we know of, because again, it can come from all these different sources, animals, whether it's mammals like mice or rabbits, or arthropods. Whether they bite you, you eat them, you come into contact with them, highly infectious. And so also, this is going to be considered a potential agent for bioterrorism, because again, it is so infectious. So that's the other criteria, right? How infectious is this going to be? Do you have to aerosolize it and contaminate everyone, or can you contaminate a subpopulation, which will then have it spread from there? Okay. Bordetella pertussis, aka whooping cough. This is preventable. We have a vaccine for this. It's a coccobacillus and very harmful to young children. It infects the respiratory system, and you get this very hallmark characteristic whooping sound of this cough, and can be life-threatening to infants and neonates. Sadly, the reservoir is us, healthy humans, that just have it, present on us, that is not infecting us. And so it transmits via contact to contact, right? So what's the first thing they recommend when a baby is born? That it gets skin to skin contact with the parent for the bonding and all that. And if that's present on the parent's skin, it can get it. It can also inhale it. That's how it gets into the lungs. So very bad outcome, but luckily, like I said, there's a Bordetella shot you can get for that. Virulence factors: it has receptors that bind to the cilia on your epithelial cells in the respiratory tract. And then it has toxins that destroy these cells. So it binds to the cilia, it secretes a toxin, it destroys those cells, and then now you have a buildup of these dead cells, which are the protective coating of your respiratory tract. And then as a result to that damage, you have a bunch of mucus that builds up, and that blocks the airways and leads to this cough. Okay. And so again, there's a vaccine, and so this really only impacts places that don't administer the vaccine, that don't have access to a vaccine, or people who don't believe in vaccines. And again, science doesn't care what you believe, right? Science is just science. I hear some screaming going on. Maybe you hear it now too. I might get interrupted here in just a second, so apologies. Would you go open the door and see what's happening? What's wrong?
Theo's gone. I didn't let him tow around my suitcase because I wasn't finished packing it up.
Okay, Theo, come here, buddy. Come here. Hey, Theo, come here. Sorry, guys. Okay. Come here. I'm talking to some friends. You want to say hello?
How come you took your phone out of your case? I tried to put it back in for you.
Yep. Okay, guys. Legionella. This is called Legionnaire's disease. And now you're calling 911. We'll let him out, would you, Bennett? Welcome to my world. This is how it is most days. Interestingly, it gets its name Legionella because it was first found in an outbreak in an American Legion Hall. And so these vets were getting together, and it was in the water. Bennett, just let him go. Okay, guys, hold on just 1 second, okay? Bennett, you want to give the lecture for me? Can I do something for you? Can I do something?
I don't know what he's talking about.
Just improvise.
Yeah, what's something you learned recently that you can tell us about?
What do you know about germs?
I know about germs that they can make you very sick. And now we had a boy alert. We had a boy alert yesterday. Or more than that. We had a boy alert. And
Okay, guys. Well, I think I might have to. Oh, is that all he wanted? Was the suitcase? Let him play with the suitcase. Goodness gracious. All right, Legionnaire disease, as I said, was named because the outbreak was first found in contaminated water in the condenser of the air conditioning units in a American Legion hall. And so the outbreak occurred in 1976, and 200 or so different members presented with pneumonia. They cultured sputum from the lungs, and that's how they identified this bacteria. And almost 30 of those 200 Americans succumbed to it. And so typically this affects males over 50. And honestly, I'm not sure where they get that statistic. I don't know if that's just because it was predominantly in this Legion hall, and that's kind of self-selecting for males over the age of 50. But it can also be a nosocomial disease that is in elderly patients that are immunocompromised. They go to the hospital for some other affliction, and because it can contaminate the water source and then be blown into the hospital room, you get this issue. If you don't have a good HVAC system, then don't do that. Stop that. Then what's wrong? Oh my gosh, we are breaking down. You know what, guys? Let's leave it at Legionella. And how many slides are left? Let's see. Okay, what do you need help with? What did you do?
my show.
Yeah, we're about halfway through. Typhoid. Oh man, there's so many good things to talk about. Okay. I'll tell you what, guys. I will leave it at this. This is all you'll be responsible for knowing up until Legionnaire's disease. And I will try to record the rest of this slide deck at a later date, and I will post it, and you can watch it or you don't have to watch it. But if you are curious about this stuff, because it's cool, then we'll go from there. All right. Any questions, guys? Sorry about that. Sorry we had to deal with that little disruption, but any questions, guys?
I do have a question, but it's more of like a one-on-one since I was still taking that exam earlier.
Okay. Why don't we just wait till people drop off and then you and I can chat? Sound good?
Yeah, sounds good.
Okay. Anything else, guys?